What's going down, Fairview? This is another episode from Mr. Knowles coming to you live from room 421, where biology is no more or no less the best. What we're going to be discussing today are the different steps and stages that take place during meiosis. We're also going to be reviewing some key terms in meiosis and discussing the idea of what does all this genetic diversity mean? We like to call this segment From Sex to Sophomores. To get started with this episode, we need to understand a few terms first before we start getting into the nitty-gritty of all this stuff. Now, what we're talking about in this feature we like to call Sex to Sophomores is the process of meiosis. Now, meiosis itself is actually the process in which our cells split to create sex cells. They are very different from any other cell in the body because we're not talking about the idea of repairing or replacing or anything else like that for our particular body, but instead creating cells where we can actually take our genetic information, independently sorted from one cell to another, and make a diverse amount of variations with our chromosomes that make us up. So, when we think about something like the process of meiosis, the true definition is meiosis is the process in which our cells divide to create sex cells, which then create 4 haploid cells as a result that are all genetically unique. Now, to fully understand this process of meiosis, we have to understand the difference between 2 hit words. 1, diploid, and 2, haploid. Now, a diploid number indicates that our cells have a full set of chromosomes. Remember the idea that with both of your parents, when you were made, both of your parents contributed equally to you. And so right now, as a human being, we have 46 chromosomes in total, 23 pairs in all. Each set of chromosomes, chromosome 1, 2, 3, all the way to 23, each set of those chromosomes all have a single contribution from both mom and then one from dad. So we end up having 23 pairs or 46 chromosomes because we got 23 chromosomes from mom and 23 from dad. This full set of chromosomes is considered a diploid number. So just think to yourself, di, like you're riding your dicycle, and ploidy, which just indicates the idea of chromosomal numbers. So we are looking at a duplicated or double set of chromosomes. Now, when we look at the word haploid, the word haploid stems from the root word haplos, which means single. So to have a haploid set of chromosomes means to have a single set of chromosomes in each cell. When we think about something like meiosis, meiosis is very different from mitosis because mitosis produces the exact replicates of our diploid cells, whereas meiosis is very different because meiosis produces 4 haploid cells with in each cell with a single set of our particular chromosomal information. So only 23 chromosomes for us. Now, one of the last things that we need to understand before we start to move into key features of meiosis is this understanding of a term called homologous chromosomes. Homologous chromosomes are chromosomes that are actually the identical genetic information for each chromosome, meaning like, for example, you both have a chromosome 6 from mom and a chromosome 6 from dad. These are both homologous pairs of chromosomes. And on chromosome 6, these chromosomes are actually going to code for these similar traits, right? Now, the deal is that we'll have then, we would call it, similar genes, right? We have similar instructions to code for a particular trait. But the cool thing is about homologous chromosomes is that now on each of those homologous sets of chromosomes, we have or can have different alleles. Now, the word allele refers to a particular version of a gene that can be different from both maternal and paternal chromosomes. So, when we look at homologs, or homologous pairs of chromosomes, we are actually looking at double sets of both your mother and your father's chromosomes that are coding for the same stuff, but the super cool thing is they can have different versions. So now that we understand the differences between the terms haploid and diploid, and now that we understand the significance of what it means to be a homologous pair of chromosomes, we need to now focus in on the steps or phases of meiosis and how they differ from the steps or phases and stages of mitosis. Now, the big deal is that mitosis is always going to start with a diploid cell. Now, that diploid cell is going to divide, and when it divides, it's actually going to make the exact replicate or exact copy of itself. Therefore, every cell that comes from a mitotic division is genetically identical. So, an organism that produces asexually is going to go through mitosis for its asexual reproduction. On the other hand, if we look at organisms that are genetically diverse, they are not copies or clones of themselves, we are looking at organisms that go through sexual reproduction. If they go through sexual reproduction, they have to go through meiosis. Now, the reason why is because meiosis, we might start out with a diploid number, but the big deal between meiosis and mitosis is that meiosis has two different divisions in it. And as that situation progresses, right, our cells, our 2N cell, divides once. But before it divided, we still replicated all the chromosomal material. So you had one chromosome from mom and one chromosome from dad, and these were homologous pairs of chromosomes, right? But they also got duplicated during interphase of meiosis. So now we have some from mom, and we have some from dad. These chromosomes can actually link together, and this is an extremely important situation because once these guys link together, they create this stuff called a tetrad. There is 4 of these particular chromosomes. So we might start with a diploid number, 2N, the full set of chromosomes that we need to be alive, right? But that number gets duplicated. And then simultaneously, once that happens, we end up going through meiosis 1. Meiosis 1 is our first division. It results in 2 haploid cells at this point in time that now have unique combinations of genetic material. Back to that in a couple minutes. Now, the second division from meiosis looks almost exactly like mitosis except for our last step. Now, the reason why is because what we're looking at that point in time is we are looking at chromosomes that are now separating, right? And so we are separating with our spindle fibers those particular sister chromatids. And as those sister chromatids line up at that equatorial plate, and as in anaphase, those spindles jump over and grab a hold of those centromeres, they tear those particular chromosomes apart, right? The sister chromatids that have been created. And they tear them apart and they put them into 4 genetically unique haploid cells. Each one of those cells has half the genetic information to create a little baby. Each one of them has their own unique allelic combinations. So instead of just having some from mom and some from dad, like in a diploid cell where you have both mom and dad's chromosome, now you might have almost an entire set of your moms, or you have this idea too of this swapping of genetic information.